Let's begin with a small but influential character in the brain's cast, Galanin. It's a neuropeptide built from amino acids, and despite its modest size, it has strong opinions about how neurons behave. Galanin is especially fond of hanging out with noradrenergic neurons in the locus coeruleus, a region deeply involved in arousal, stress, and attention. When Galanin speaks, these neurons tend to calm down, which is helpful when the brain has just a bit too much excitement. Galanin is far from alone. The nervous system communicates using an impressive vocabulary that includes classic neurotransmitters, gasotransmitters, and neuropeptides. Substance P, for example, is famous for its role in pain and stress signaling. GABA, an inhibitory transmitter derived from glutamic acid, acts as the brain's brake pedal. Glutamate, by contrast, is a primary excitatory signal, while glycine plays a similar inhibitory role in certain circuits, especially in the spinal cord and brainstem. Then there are the gases. Nitric oxide and hydrogen sulfide may sound like lab hazards, but in the brain they function as gasotransmitters, diffusing freely across membranes and influencing nearby neurons without knocking on the door. They add a certain informality to neural communication, drifting in, making a point, and drifting back out. Neuropeptides operate on a slightly different timeline. Unlike small molecule transmitters, they are synthesized as pre-peptide precursors in the cell soma. These long protein chains are trimmed and refined by convertase enzymes as they travel down axons and dendrites. Once ready, they are packaged into large, dense core vesicles, waiting patiently near the nerve ending for the right moment. When that moment arrives, calcium enters the neuron and sets exocytosis in motion. Proteins like soluble N-ethyl-melamine sensitive factor and calcium-dependent activated protein for secretin help vesicles fuse with the membrane and release their contents. Compared to fast synaptic transmission, neuropeptide release is slower and more deliberate, more like a policy memo than a text message. Once released, neuropeptides usually act through G-protein coupled receptors. These receptors don't just flip switches, they initiate cascades that reshape cellular behavior. Galanin, neuropeptide Y, cholecystokinin, and tachykinins, such as neurokinin B, all use this strategy. Neurokinin B signals through the NK3 receptor and plays a role in reproductive hormone regulation and stress response. It's a reminder that even the smallest amino acid-based signals can have outsized effects, quietly shaping how we feel, respond, and adapt in an ever-changing environment.